Right. I'm Toby Bowser. I'm a professor in the Department of Medicine. I'm our imaging board for the Wisconsin University. So I'm just going to go over a lot of things you've already seen, but I'm going to go into a little more depth into specific imaging things that we do here at Wisconsin, some of the facilities we have, and what it takes to do an imaging study, and then uh, kind of end with kind of an intro to like what we're going to get. So disclosures. So I'll spend kind of the first of just talking about prime pet imaging ADRD, some of the specific signatures we get from these modalities, and then Wisconsin. So you've probably seen some of these already, but just kind of to get you acclimated to what we expect to see in Alzheimer's disease and neurodegenerative disease or in general. These are different MRI sequences, right, that we can set up, and we can acquire these within, say, 30 minutes to an hour on a, single. And so kind of our main, bread and butter sequences are going to be things like T1-weighted MRI and PT-weighted MRI. These primarily give us information about structure, right? So we can get things like gray matter volume, white matter volume, volume of CSF. And the idea in these neurodegenerative diseases, we expect to see brain atrophy, so we expect to see CSF volume increasing and taking up the space from the atrophied brain. And coincidentally, we also see things like reductions in gray matter volume. Right. So these are kind of our neurodegeneration biomarkers. And this kind of on the left here shows kind of the expected direction that we would see as disease progresses, right? So increase in spinal fluid volume, decrease in gray matter volume. And another thing to point out here is thinking about co-pathology. So there's kind of, they're not super diagnostically accurate, but there are attribute signatures that are common in different neurodegeneratives. So in Alzheimer's, you expect to see temporal atrophy. In TDP late, you might see more hippocampal atrophy relative to other regions of the brain. And so these kind of regional patterns of atrophy give you some indication of underlying etiology. By themselves, are probably not accurate enough to be like that. So that's mainly kind of our structural measures. And on the right, this is a small arsenal all day of tools that we have for vascular imaging. So kind of the main staples that we see in our field are T2 flare imaging, where we see things like white matter hyperintensity, ischemic lesions in the brain. This flare imaging is also becoming really important because it's how we monitor for amyloid-related imaging abnormalities, which is ARIA, in anti-amyloid treatment. So if you've heard, kind of brain swelling, edema, these kinds of things associated with those treatments, this modality that we use to measure those things. Other things we can use with T2 star and susceptibility rated imaging, we can see kind of these things that look like little holes. I promise you these holes are not actual holes in people's brain. It's artifact, potential artifact, right? So what happens is hemocysteine deposit that has a small amount of ferrous material in it, and this creates an artifact on MRI. Basically makes things like bigger than they are, but they're really nice for detecting kind of these small microbeads in an MRI. And so like things where people have a cerebral amyloid angiopathy, you might see a lot of these kind of in the posterior part of the brain. Then lastly, for the vascular MRI today is arterial spin labeling. This is more of a measure of perfusion. So again, we expect the perfusion deficit as the disease advances. And another thing that I hope be useful for, and typically other modalities, is also in stroke, right? So again, so you can really clearly see, for example, if a region doesn't have blood getting to that part of the brain, so it's pretty diagnostic so shifting over to PET, you've seen this, I think, 4 times now, but I'll give you a little bit more context about how to interpret these things and what we get out of amyloid and tau imaging. So the idea with these PET scans and the way that they're designed is they're kind of receptor ligand binding interactions, and they're displaceable. And by using some kinetic models and simplified approaches, the measures that we get, in terms of like standard uptake value ratio, unemployed, or distribution volume ratio, all of those things represent the same thing, which is they're giving you a measure of kind of the density of whatever that protein is in that region. The other thing to know is that these are measuring insoluble aggregates, right? So Jeff in his pathology component after this, we'll talk more about these, but amyloid and tau start off as these like clumps of proteins, right? They undergo kind of this whole progression of going from soluble toxic forms, and then start self-aggregate. And so aggregated forms will appear. And so kind of putting all these things together, this is kind of going from the back to the front of the brain. We get these nice three-dimensional pictures where we can see things like structure and atrophy in the image on the left with our T1 MRI. The regions that are more warmer colored, like yellow, orange, red, those are showing higher density of those proteins. So tau in the middle and beta amyloid on the right. So these things have different reasons or different utilities in research. So for tau imaging, the spatial component is a lot more interesting and a lot more associated with tau spreads kind of outside of the temporal lobe into the neocortex. We see increasing levels of dementia, etc. So beta amyloid is a bit more diffuse and kind of goes across most of the neocortex. There's a little bit of staging that, like, once people get kind of closer to the dementia phase, they see a lot more posterior amyloid measured like that. And then you see these curves already, so I won't spend too much time on them. But again, just to kind of straight home that these aggregation processes measured by PET really follow these really predictable patterns. The plot on the left is some current work that we're doing in my lab. We're looking across 3 different cohorts. Actually, 4 cohorts we've done since I made this slide, but essentially showing that these things accumulate at a very predictable rate. In sporadic AD, you do see some population effects in groups like people with autosomal dominant AD or people with Down syndrome. These are genetically linked mechanisms that implicate A-beta production. Another kind of place that we're going with these types of things is by being able to find, when the processes start, trying to understand how predictive they are of dementia, and the y-axis here is mislabeled, should be dementia-free probability. So everybody that loves Kaplan-Meier analysis can grow up at some point. But the idea, right, is that as we have amyloid and tau for longer and longer periods of time, we see the rate of dementia start to increase, right? So, dementia in this case would be one axis, but the other thing, just in terms of the timing of disease, right, we really don't expect people to make it much beyond, say, 25 years of amyloid positivity or about 15 years of tau positivity before they start to see, before they reach the stage. And another thing that we don't do as much of here, but another tool for PET imaging is fluoride oxy glucose. So this is a tracer that basically mimics glucose, so it gets metabolized in the brain. And just because of where it's radiolabeled, it actually gets kind of stuck in a part of that glucose cascade. So essentially what you see in neurodegenerative diseases is a reduction in glucose metabolism, right? So neurons and things are dying. You start to see this kind of lowering of glucose metabolic activity in the brain. And this is another case similar to MRI, where there's kind of spatial signature. Here's a paper from Children's group who looked at differentiating. They had neuropathological diagnoses on these people before or after they died. They had before, and just showing some of the differences in the spatial patterns that they see between 80 versus controls or 80 versus 8 and these kinds of pathological pictures. And then one last thing that we've recently started, and I will also touch on, is we started doing ex vivo analysis for our brain donors. We take a fixed hemisphere of the brain, cerebellum and brainstem, and actually image that on a standard Weissman. And we're using this to get kind of spatial concordant comparison of antemortem imaging and actual MRI. And so this is some ongoing work and new things that we've started doing. And then this kind of gets acclimated to our facilities here. This is kind of a map, and right now we are in the HSLC, so kind of over here. The types of things that we have on site are, we have an on-site cyclotron lab or a giant physics. So they handle things like making radioactive coats that we attach to our PET tracers. They also do some radio tracer production and have applications far beyond just neuroimaging and neurodegenerative disease. The radiology group also has an Orion PET scanner that we use for certain studies. And then we do a lot of our imaging at the White Center, which is across the road, maybe familiar with. But there we'll see a radio pharmacy production lab, some of the PET systems there as well. And then this is just to kind of give you a sense of like for PET imaging, why this is so expensive, and why it takes so many people. So again, just to be able to make a tracer on site, but if we have something that's carbon 11 labeled, the half-life of carbon 11 is 20 minutes, so you can figure about an hour and a half everything that you made is gone. So you have to be able to make that radioisotope tracer and get it injected and get it scanned in a pretty short amount of time. There's advantages to this. We get lower dosimetry for radiation and things, and we can do like say an amyloid scan or tau scan the same day. That radioactivity. But in order to do this, we have to have a cyclotron, which is over $1 million. It takes 2 to 3 people to like maintain and operate. We have to have radiochemistry facilities where we actually can take the radioisotope, attach them to the chemical matter, clean all of that up, and be able to get it to the point where we can inject the humans. So that also requires radiopharmacy. Do this all in a sterile environment. And then that's making a tracer, then we also have to inject it, which takes PET technologists, we need PET scanners, and then that's just getting data in the door, and then we have to have a whole informatics platform to be able to process and analyze images. So just in collecting a PET scan, there's probably 10 or more people involved, or scheduling all the way through the pretty big operation. And then this is my last slide. So this is our imaging core at the Alzheimer's Disease Research Center, and some of the services that we provide. We currently support over 13 studies, including the cohorts you mentioned, so WLS, RAP study, or ADRC, MIDAS. And then our core provides many services, so like Rachel mentioned, we're very collaborative, very helpful, pre-award, post-award consultation. If you're interested in anything imaging, but you're not quite sure maybe what things to utilize. And with Rachel, we also usually kind of have joint consultation of like, are there kind of combined approaches to using imaging and biopsies that help your budget and but still makes sense in terms of perspective. And then I think the last thing I'll mention is just we've scaled this up quite largely. Certainly, obviously, to be part of this, Brad and Andre. But today, I just looked at this morning because I wanted updated numbers, but we have over 6,500 MRI scanners across all the studies we support, over 2,600 amyloid PET scanners, so it's a pretty massive resource. And as far as longitudinal data goes, it's probably one of the most comprehensive in the world in terms of amyloid packaging. That's all I have. Thanks.